you for coming tonight. Many of the donors and supporters in this room are vital to supporting our programs, and I want to recognize these programs tonight. Our Patterson Scholarship Program, and Libby Patterson, thank you so much for your support of this program. Many people in this room are supporters of our Hope for Reefs initiative, and I thank you very much for that, and you'll be able to hear more about the impact of your support when Rebecca speaks, so thank you very much. And finally, I'd like to thank the Claire Matzker Lilienthal Distinguished Lecture Series supporters, Lucy Matzker and her family. Thank you so much. So I want to talk very briefly about the Patterson Scholar Program because it gives me great pleasure to announce that Rebecca Albright is our newest Patterson Scholar in the program. So congratulations, Rebecca. This is a very innovative program. It was really thanks to the generosity of the Academy's board chair, Libby Patterson, and the Patterson Scholar supporters, of which there have been many, to support this program in what we call the Patterson Scholars of Science and Sustainability. And these are endowed chairs in honor of the late William J. Patterson, who was chair of the Academy's Board of Trustees from 2007 and 2010. To 2010, and he knew us very well, and he knew what was really core to what we wanted to do with this great institution. So the Patterson Scholars, it's a very innovative program. We are science educators of exceptional talent, achievement, and productivity that are committed to exploring the natural world, but also addressing the challenges of sustaining this natural world and being innovative science educators and communicators. So you'll see that Rebecca personifies all of these traits. I won't give it away. I'll stop. So the other program that we're here to celebrate tonight together is the Lilienthal Lecture Series. So the Lilienthal Lecture Series is a program that ties effectively science communication about important science outcomes, so it's authentic science communicated with passion. And again, you're going to see some of the impacts of science, excellent science communication through Rebecca's presentation. So together, the Patterson Scholar Program and the Lilienthal Lecture Series and Hope for Reefs, as you can see, is really trying to move the needle through both innovative, authentic, excellent science and excellent science communication and engagement. And I just wanted to draw out some examples of some of our excellent Patterson Scholar impacts. Not only is Rebecca here tonight, but we have our inaugural Patterson Scholar, Brian Fisher, who, can you raise your hand, Brian? He's in the back. So Brian Fisher began his research on many fronts, but he's made incredible research gains in understanding the biodiversity of native insect fauna in Madagascar, and he's worked very hard to find that sweet spot where we can both preserve natural systems and also benefit humans that live together with those natural systems. So he's developed an amazing program to find solutions to malnutrition through providing scaling, identifying scaling, and providing proteinaceous sources from crickets, and apparently they taste very good, to people in Madagascar that would normally hunt bushmeat, lemurs, and otherwise degrade the natural environment. So it's a wonderful win-win. And he is also an excellent science communicator. And then we have another one of our Patterson scholars here, Scott Laurie. Also in the back. And Scott is co-director of iNaturalist. So who uses iNaturalist? Okay, you're not allowed to leave the room until you sign up for iNaturalist. So iNaturalist is an amazing platform to observe biodiversity on our planet through the assistance of citizen scientists that go out and document natural history with their phones. It's evidence-based, it captures a picture, and it connects them to a world of experts that can then help them identify that. And those observations get logged so that we can all use it to really drive scientific discovery, but connect it back to the people that provided those observations. So it's a really innovative program, a citizen science program. It's the top citizen science program, arguably, in the world. It's the 25th most accessed website. So it's grown phenomenally, and it's going to change the world. So thank you, Scott. I'm also a Patterson scholar. Thank you. And I spend a lot of my time working on viruses that occur in mosquitoes, and I'm really trying to understand how the erosion of biodiversity might propel viruses into human populations, whereas biodiversity normally could protect us and mitigate from infectious disease emergence. So more later. With that, I just want to say a brief thank you to the Lilienthal and just give you a brief overview of the Claire Matzker-Lilienthal Distinguished Lecture Series. This is supported by a generous endowment established by the family of the Claire Matzker-Lilienthal, Claire was a remarkable leader of many civic, educational, and charitable organizations. She served on the San Francisco Board of Education and the Board of Trustees of the California Academy of Sciences. And to honor Claire's passion for education and inspiring science communication, the Academy hosts a lecture each year, and we are thrilled that this year's Claire Matzker-Lilienthal Distinguished Lecturer is Dr. Rebecca Albright, who is also our newest Patterson Scholar. Okay, hold on. Sorry. We're almost ready. Now I want to introduce Rebecca. Okay. So Dr. Rebecca Albright is Assistant Curator of Invertebrate Zoology and a Patterson Scholar at the California Academy of Sciences. She is a coral reef biologist with expertise in coral reef biology, ecology, and biogeochemistry. You'll find out what that is in a minute. Her research focuses on the ability of coral reefs to cope with changing environmental conditions, such as ocean acidification and warming. Since joining the Academy in 2016, Rebecca has increasingly focused her efforts on solutions to the coral reef crisis, including the Academy's commitment to scaling coral reef restoration. She has worked in government, academia, and nonprofit settings, and has studied coral reefs around the world, from Florida Keys to the Great Barrier Reef. In addition to her commitment to research, Rebecca's passion for community engagement has been fostered by years of working with nonprofits and citizen science organizations that focus on educating communities about coral reef conservation and management. So in this talk, you'll see Rebecca give you an overview of some of the challenges that reefs are facing and how she's going to address them. And we're so pleased that Rebecca is part of the Academy family, and I want to welcome you to the stage, and thank you so much.
Thank you, Shannon, and thanks to all of you for coming tonight and for being so passionate about the plight of coral reefs right now to share your Tuesday evening with me. I am really excited about this talk tonight. A lot of you know that coral reefs have been in the media a lot over the last couple of years, and arguably for all of the wrong reasons. And one of the reasons I'm so excited, not just because it's such an honor to be a Patterson Scholar, but because for a couple of years recently, it was really hard to stand up as a coral biologist in front of a group of people and talk about optimism and hope because of everything that was happening. And I can honestly stand here tonight and look all of you in the eyes. I can't actually see you because of the lights, but if I could, I would look you all in the eyes and I would tell you that I do have optimism and I do have hope, and I'm hoping that I can share some of that with you tonight. So thank you for being here. So when most of us think about coral reefs, we might picture something like this. We might picture some aesthetically beautiful, vivid picture that of a place that's teeming with life, colors, fish, all sorts of different organisms. We might picture a couple of charismatic megafauna, some sea turtles, a coral reef, maybe a shark, Nemo, some brightly colored corals. Or we might picture a really nice tropical vacation somewhere, maybe in Australia, that all of us might like to be, and maybe not in the chill weather of San Francisco that's happening right now. But what you might not think about is all of the ecosystem services and value that coral reefs actually offer us every single day, free of charge. So coral reefs actually cover less than 0.1% of the ocean floor, but they host almost 30% of biodiversity in the oceans. So they're really punching above their weight class in terms of being hot spots for biodiversity. We don't call them rainforests of the sea for no reason. There are also over 200 million people around the world that depend on coral reefs for coastal protection and their own livelihoods. It's estimated that a square patch of reef that's around 5 meters squared will mitigate up to 97% of wave energy and when a storm is coming through. And there are places around the world, like in the Maldives, where you've had coral reefs or you've had sand that's been dredged out, and it can cost almost $10 million per kilometer in order to restore a seawall to recreate these natural protective services. Coral reefs also provide the primary protein source for up to 1 billion people around the world by offering 5 to 15 tons of fish annually per square kilometer. And they give us almost $36 billion a year in annual tourism revenue because we love to go and snorkel and dive and visit these beautiful tropical locations and spend our leisure there. And if you wrap all of those ecosystem services up and total them, it's almost $400 billion a year that these systems are offering us free of charge. It's a human security issue, it's a livelihood issue, it's an aesthetic issue in terms of why we value and love these ecosystems. And so I want to take a minute and just back up and talk about what a coral is and what a reef is, because a coral is not a reef, just like a tree is not a forest. And so I get asked a lot this question of, is a coral an animal, a plant, or a rock? And the question is yes, or the answer is yes. And so if we look at what a coral is, this is one coral, and what it is a calcium carbonate skeleton, so it's basically a rock that is coated with a living thin veneer of animal tissue. And so they're very fragile, and so this animal tissue covering a rock, and inside of the animal tissue is an algae. And that algae photosynthesizes and provides the animal with almost 90% of its food requirements. And what you can see by looking at this coral up close is that it's made, they're clonal organisms, they're colonial organisms. It's made of hundreds to thousands to millions of tiny little individual polyps that all communicate with one another and share resources with one another in ways that we don't even fully understand. And so I want to take a look at how this happens, just so you appreciate how a coral becomes a reef. So if we look at one individual polyp, which might be a couple of millimeters across, depending on the species, it looks like a baby sea anemone, and it has a mouth, and it has a gut. And these animals can actually eat plankton. They'll actually capture things from the water column, pull it into their mouth, and digest them. But they also have this algae that looks like this. It's a single-celled algae that lives inside of the animal tissue and provides it its color and also provides, in some cases, as I mentioned, up to 90% of the food. When we think of coral reefs, we think of these clear, beautiful waters you can see for hundreds of meters when you're diving on them. And the reason they're so clear is that there's not that much in those waters in terms of nutrients. So in terms of food for the reef, corals have evolved over evolutionary time periods this amazing symbiotic relationship with an algae that lives inside their tissues and provides them with energy. They're also coated with microbes. And we're still underappreciating this relationship as well, just like we're starting to understand the relationship between our gut microbiome or our microbiome and how it influences our overall health and physiology. We're just scratched the tip of the iceberg in terms of understanding how the microbiome of the coral influences its physiology. So what we actually call this is a holobiont in the scientific community. So the coral is part animal, part algae, part rock, and part microbe. We call it a holobiont, and it's all those different things interacting together. So how does this take place? First of all, a coral starts its life as a tiny little larva. It looks like a sesame seed, might be a millimeter across, very, very tiny. You might mistake it if you're trying to find it with the naked eye, and they swim around. You might have baby pictures at home. I have a baby at home, but these are pretty cute. And they are coated with cilia. So these little tiny sesame seeds are coated with cilia all over their body. The cilia beat up and down their body, and they rotate. And they swim around like little tiny torpedoes looking for places to settle on a reef. And you can imagine that that's a pretty deterministic and important decision to make, because corals may live hundreds or thousands of years. And so once it settles onto a reef, it's setting up shop for a very long time. So these seemingly simplistic organisms are actually very sophisticated in terms of their ability to be able to sense chemical cues in their environment. This tiny little larva has an oral end and an aboral end. The oral end is used for taking up food, and the aboral end is chock full of chemoreceptive and light receptive or photoreceptive cells so that it can sense its environment and figure out where's a good place to set up shop. Once it finds a good place, that larva will metamorphose like a caterpillar into a butterfly. So it actually, the aboral end flattens out and cements itself to the substrate. The oral end is up, and it starts to differentiate into tentacles. Those tentacles will then become the mouth and start to pull prey into its mouth. So these baby corals look like they might be about 24 hours old. They haven't started calcifying yet, but they've metamorphosed and they've set up home. Around 36 hours later, it'll start to calcify. So you can see the primary polyp there, and you can see the evidence of some calcium carbonate skeleton there underneath the tissue. That primary polyp will start to bud new mouths. Asexually, they start to bud new mouths and grow laterally and vertically. And they start to take up the symbiotic algae. So you can see it's starting to brown out a little bit. This happens over the course of several weeks. And this is actually an individual that's back of house right now because we're growing up coral babies right now. Same thing from a side view. So this again is probably, this guy's probably about a month old at this point, but still only millimeters in size. This guy's probably a couple of months old, and he's started to bud. This might be several larvae that fuse together, but it's starting to grow and take up more algae. And over the course of years, this will grow into a coral colony. So this is a colony of Acropora hyacinthus that is probably somewhere in the range of 3 to 5 years old. Over time, you have multiple species, multiple colonies doing that, and you get a reef. This is probably 10, 20 years worth of growth right here. Excuse me. And then over geologic timescales, you get structures like the Great Barrier Reef that you can see from outer space. And the fact that these things start out the size of a sesame seed, tiny little larvae, and can grow living structures the size of the Great Barrier Reef, 2,300 kilometers long, that you can see from outer space, continues to blow my mind, and I've been studying them for over 20 years. And you would think that with a system this valuable that offers us almost $400 billion free of ecosystem services every year, human food, livelihood, coastal protection, biodiversity, and is this long-lived, that we would do everything to try to protect these systems. And unfortunately, we're not. So coral reefs are in trouble. You've probably seen this all over the news recently. It's estimated that we've lost about 50% of corals worldwide over the last 30 years. And we're projected to lose 90% of what's left by the middle of this century. So let's take a quick look at why that's happening. We have a variety of different threats on coral reefs, from local threats to regional threats to global threats, and we're going to talk a little bit about each. In terms of local threats, we have things like development. So this is literally an airstrip that's built on top of this coral reef, off of an island. This is all a fringing reef with an airstrip right down the middle. We have coastal local communities that depend on the reef for fishing and their livelihoods, but unfortunately that does sometimes put a lot of pressure on the reef system in terms of nutrient pollution, overfishing, and extraction. Here's another example of fishing that can be done at a local level and also at a commercial level. And we have pollution on reefs, which you think might be concentrated to the shallow waters, but our deep diving team has found evidence of pollution in remote coral reefs all the way down to 50, 100 meters. So we have flip-flops, fishing line, plastic bags all over the reef. We've seen diapers, you name it, you see it. And there's a little bit of out of sight, out of mind that people think, well, I don't actually see the reef, I can't see what's under the ocean, so I'm probably not impacting it. But once you get your eyes on it underneath the water, you see all sorts of human impacts on these systems. On top of the local threats, we have global stressors.
So this was taken just a couple of days ago, May 18th. This is a reading of the most recent atmospheric carbon dioxide levels taken at Mauna Loa, which is a research station in Hawaii. And they've been monitoring atmospheric carbon dioxide levels at this research station since the early 1950s, or the late 1950s, sorry. And as you can see, when they started taking these measurements, the atmospheric carbon dioxide levels were in the low 300s. And over the last 50 to 60 years, they've crept all the way up over 400 parts per million, and now, as of last week, over 415 parts per million. And the reason why that's significant, you might have seen it in the news, is that this is the first time in human history on this planet that CO2 levels have been over 415 parts per million. In human history. And the oceans do us a huge service in mitigating the results of that, or what could be the effects of that on the planet. So the oceans cover over 70% of the planet, and water has a high thermal capacity. So it's estimated that the oceans actually absorb 93% of the heat that would otherwise be emitted into the atmosphere by all of these carbon dioxide and greenhouse gases that we're emitting every day. 93% is taken up by the oceans. So this is a map of temperature anomalies that have occurred over the last 100 years or so, since before the Industrial Revolution till today. And so you can see that on average, the oceans have warmed around 1.8 degrees Fahrenheit, or about a degree Celsius. Since the 1970s, there have been half a degree Celsius of warming that has occurred. If the oceans hadn't taken up 93% of that thermal energy, it would not be half a degree. It would have been 36 degrees Celsius that the atmosphere would have warmed. That would render the entire planet inhospitable, or inhabitable, also inhospitable. And so they're offering us a huge service in terms of climate regulation, but this does not come without a cost. And so unfortunately, we're seeing the effects of all of this thermal warming on ecosystems and organisms all around the world. You've seen pictures like this time after time in the news over the last couple of years of formerly healthy reefs that are now entirely bleached. Not just a couple of corals here and there, but the entire system turns white. And this change can happen over the course of a couple of weeks, which is frightening. Here's another reef. And these pictures were actually taken and sent to me a couple of days ago by our team, our ichthyologist, Luis Rocha. We have a team of deep divers right now in French Polynesia. They're in Tahiti. And they were there 8 weeks ago, and the reef looked like this. And then they were there this week. And that same reef, this is the exact same reef within an 8-week span of one another. So just to talk about what bleaching is for a second, because you've heard a lot about it, we'll talk a lot about it in this talk. So what is it exactly? So you have a normal healthy coral, which we've already talked about, is an animal with an algae that lives inside of its tissue and provides it with food. Under stress, which can be a variety of different stressors. It can be cold stress, salinity stress, heat stress. Right now, the big elephant in the room is heat stress because of global warming. Under stress, what we think happens, the mechanism of this is still not completely understood, but what we think happens is that the algae get overreactive, either because of too much light or too much temperature, and they start producing reactive oxygen species like free radicals and things like that that are harmful to the animal. And the animal can handle a certain amount of that, but once it starts to get too much, the animal cannot handle it. And so what it does is try to get rid of the algae. Much like you would if you had a bug in your system, you got food poisoning or something like that, you're gonna do the fastest thing to get that out of your system as a last-ditch effort for survival or health, and you'll purge it. And that's what this animal does. And so it purges the algae out of its system. And what it's just done is gotten rid of its primary food source. The animal's not dead at this point. A bleached coral is not a dead coral. It can recover. And so what it looks like is you see the brown algae's left. So you see a calcium carbonate skeleton with a thin veneer of tissue, transparent tissue over top. And it literally looks like you've taken it and dipped it in bleach. That's why we call it bleaching. But it can recover if the stress is removed within a certain amount of time. So that's really positive news. But the problem right now is we're not removing the stressor. And if that doesn't recover in a certain amount of time, the tissue will eventually die and slough off. The rock is left, and then that rock can get overgrown with other algae and things like that and turn into a dead coral. So bleaching happens all the time in patchy locations because of localized stressors. You might have an extreme rain event that causes a salinity fluctuation, and you get a certain area of corals that bleaches and then recovers later. But right now the concern are these global mass bleaching events that are happening where the whole world is affected simultaneously, and that shouldn't be happening. And the reason it's happening is global warming. We've had 3 global mass bleaching events ever. The first one was in 1998. The second one was in 2010, and the third one started in 2014 and lasted, we thought it would last just a year, like most of them do, and it just kept going. And it actually lasted 3 years, from 2014 to 2017. It was the widest spread and most catastrophic mass bleaching event that's ever happened. Over 70% of reefs worldwide were affected. What this map is showing, so NOAA has what we call a coral reef watch, where they will map out temperature anomalies around the world and show what projected temperatures are so that we know whether areas are at risk for bleaching. And they will release these bleaching alert levels. They'll say, well, this area is a bleaching warning, or alert 1 or alert 2. And what you can see is over this 3-year period where we had this third event recently, there is nowhere in the world that escaped that event. 70% of reefs worldwide. We've never seen anything like that. So these bleaching events are now occurring 5 times more frequently than they did in the 80s. That's a really quick change over a short period of time. They're happening about every 6 years as opposed to every 30 years. And the problem is because corals are slow-growing organisms, you saw that coral at the beginning that was maybe this big and was maybe 3 to 5 years old. They're slow-growing, so in order for a reef to recover, it needs around 10 to 20 years. And if you have mass bleaching events every 6 years, obviously that's not happening. So the frequency of these acute events is increasing, and the recovery time is narrowing such that they're never really getting the breathing room they need to recover. Now, to add insult to injury, not only does the ocean take up thermal energy, but it also takes up CO2 directly. So again, we have that 70% of the globe covered with water, and at the air-sea interface, you just have gas exchange, like you would anywhere across sea and water. And so across that 70%, you have the oceans, the surface oceans, just taking up CO2 because of gas exchange. And it's estimated that the oceans have taken up about 30% of the CO2 that we've released into the atmosphere. And that causes what we call the other CO2 problem. And we call it the other CO2 problem because you can't see it. It's not visual like bleaching. It's much more insidious. But it's ocean acidification. And this is one of my areas of expertise. And what ocean acidification is a shift in the carbonate chemistry of seawater over time. And the result of that is such that it makes it much more difficult for many calcifying organisms to secrete calcium carbonate or any sort of calcification for a supportive structure or skeleton, for either protection or support. So these chemical equations are not really that important, but we're a scientific audience, so we'll just go through them really quickly. So when CO2 reacts with water, it creates an acid, and that acid's called carbonic acid. And it's very weak, and all that means is that because it's a weak acid, the ions that make it up don't hold together very strongly. They dissociate pretty easily. And so when they dissociate, you're left with hydrogen ions in the water. And those hydrogen ions that are free start to slowly change the pH of the water. And in order to maintain homeostasis or try to maintain some sort of balance in the ocean, you have naturally occurring ions, like carbonate, that will go and try to bind up that hydrogen so that the pH doesn't change. And so what we get with ocean acidification is an increase in CO2, an increase in your hydrogen ions, and a decrease in carbonate. And the problem is that for anything that secretes calcium carbonate shell or skeleton, less carbonate means it's much more difficult to secrete that skeleton and create a structure. And so what this map is showing you, all of the pink dots here are coral reefs all around the world. And the colors here are showing you a value called the saturation state, which basically, for the sake of tonight, it's just how easy it is for something to calcify. And so the more blue it is, it's much easier for reefs and any other calcifying organism, a lobster, a clam, a crab, anything that calcifies, to calcify at this end of the spectrum as opposed to this end. So the atmospheric CO2 was 280 about 100 years ago before the Industrial Revolution. Creeped up towards 380. You saw at the beginning of the talk we're now around 415. And then as we move into the future, this is what happens to the oceans in terms of their acidity level and the amount of corrosive water that's there. And we're projected to be around 750 by the middle of this century, not the end of the century, the middle of this century. And so the concern is that you can imagine is that for a reef, not a coral, but for a reef is this geological structure that was built over thousands of years, and it was this constant balance between constructive processes of calcification by all of these tiny little organisms over time against destructive processes of wave erosion and physical mechanical breakdown. And in order for the reef to grow through time, obviously constructive has to outweigh destructive. And the concern is that ocean acidification is reducing construction, increasing erosion, and it's going to shift these systems into an actual state of net loss. So you might have seen headlines because of papers that have been published that say coral reefs are going to be dissolving by the middle of the century. It's not that the reef is literally going to be bubbling away in front of your eyes. That's not going to happen. But it's that over time, these systems are in a state of net loss. They're not actively growing. So with all of these things taken together, the United Nations has a group called the Intergovernmental Panel on Climate Change, or the IPCC. And last fall, they convened and released a report saying, what are the consequences if we approach 1.5 degrees warming? Bear in mind that we are now on target for 3 to 5 degrees warming Celsius. The only way, we've already had 1 degree. The only way that we're going to get 1.5 degrees is if we stop all CO2 emissions tomorrow, and we have to actively sequester CO2 out of the atmosphere. We have to pump it out and store it somewhere. That's the only way we're going to do this. So what's the result if we are able to do that at 1.5 degrees versus go towards 2 degrees? And they looked at all sorts of different ecosystems, but for coral reefs, which are one of the most vulnerable to these changes, if we're able to cap it at 1.5, we risk losing 70 to 90% of coral reefs around the world. And if we edge towards 2 degrees, we effectively lose everything, upwards of 99% or more. So, at this point in the talk, you are probably wondering why I said there is hope. But I do believe there is some. We are not going to save everything. That ship has sailed, but we can save things. And there is a shift in the community that is palpable right now that is very energizing to me. And I wouldn't tell you that if I didn't believe it, because trust me, this is something I think about every day. So what can we do about it? We are at a point right now where the community is shifting. It's changing because people are realizing that we're running out of time. And the necessity is the mother in invention. And there are some really exciting changes in terms of the way people are thinking about things, the collaborations that are happening, the focus areas that people are kind of putting all of their energy into that have happened over the last 5 years that have never happened in the community before. And we're not there yet. We don't have a solution yet. And there is no singular solution to this. But we're headed in the right direction, and I think that we're on the verge of making some really positive change because people are looking in the right areas. So as a couple of examples, last December I attended the first ever Coral Reef Symposium that was focused solely on intervention science. So the coral reef scientists get together every year. We get together notoriously. There's this huge coral reef convention every 4 years, and this has happened for decades. And we talk about biology and ecology and everything that's wrong and what's happening. Never really do we talk about solutions. This is the first time ever that everybody around the world got together in Florida and talked for a whole week about what do we do about this. So this is one example of change that's starting to happen in the community. The National Academy of Sciences released a report, over 400 pages last fall, that was a consensus study looking at interventions to increase the persistence and resilience of coral reefs. 400 pages worth of stuff of people trying to pull together their best and biggest ideas of what do we do, how do we do, when do we do it. And it looked at everything. We're going to talk about some of these, not all of these, obviously, it's 400 pages long. But it looked at things like managed breeding, managed selection, cryopreservation, phage therapy, antioxidants, managed relocation. Can we take corals from the Red Sea where they've seen warmer temperatures and move them to the Great Barrier Reef and replant the entire Great Barrier Reef with Red Sea corals? Can you shade coral reefs and so they're not getting the light stress and the temperature stress? can we offset ocean acidification? This is one that I actually led, which is, can you just dump a bunch of soda lime in the water and neutralize ocean acidification? You can do that. It's not scalable, and it doesn't last very long, but you can totally do it. Seagrass beds. Can we plant a bunch of seagrass, or this is called phytoremediation, things that photosynthesize to suck all the CO2 out of the water? People are trying to hit this from all sorts of different angles, and that's never happened, and we're only 5 years in, and so I feel like it's only a matter of time before we start to really affect some positive change. So I'm going to just talk about a couple of these that you've probably heard about that have gotten a lot of attention. And then I want to talk about what we're doing here at the Academy and what our niche is and our angle is and how we feel like we're contributing in a really meaningful way to this new focus area. So supercoral. You guys have probably heard about supercorals or frankencorals. And the idea here is, can you make a coral, manipulate a coral so that it miraculously survives heat stress, so that it's bleaching tolerant and bleaching resistant? So how would you do that if you wanted to do that? There's a lot of people trying to attack this idea right now. So remember that the coral itself is part animal, part algae, and part bacteria. And so you can tinker with each parts of this in order to see how it would affect the whole organism. And that's what people are doing. And what you might imagine is that certain parts, like the animal itself, has a very long generation time. The best case scenario for an entire coral is from birth to creating another animal in 4 years. So trying to selectively breed something that's that slow when we're losing entire swaths of reef over the course of a couple of weeks, not super practical. But the algae, or the microbes, have a much shorter generation time. You can evolve those in the time span of a couple of days or a couple of weeks. And so could you tinker with those in a way that meaningfully helps the overall animal? So there's a few different ways that you might do this. One is through breeding. One is selective breeding. So after a bleaching event, you might have two corals next to one another, exact same species, different individuals, one bleaches, one doesn't. Why? Genetic variation, and we don't really understand it at this point, what it is that's inferring resilience to that one coral. But much like there's genetic diversity in this room, and some of you might be more vulnerable to disease than others, or some might get the flu if it swapped through and others don't because of their immune system. Corals are exactly the same. And so after a bleaching event, if you can go out and collect the survivors and preferentially breed those, then you might be able to somehow outplant bleaching tolerant corals. Another way is by creating hybrids. People are actively working on this in Australia. There's something known as hybrid vigor, where if you create a hybrid out of two different species, that hybrid is more tolerant to stress than either of the individual species. And so people at the Australian Institute of Marine Science are breeding species together to create hybrids and then thermally testing them to see whether they've actually inferred thermal tolerance to these individuals and if they would be good candidates for reef restoration work. Another thing you could do would be manipulate the microbiome. So we do this all the time. We take probiotics. There's all sorts of things that we're doing right now in human health to try to manipulate our microbiomes to optimize health. And it's the same idea with corals. People are trying to understand what is the composition of a coral microbiome, what are candidates that make it really healthy, and can we preferentially culture those strains of bacteria, and then after a bleaching event or before a bleaching event, go out and sprinkle coral probiotics on a reef and get them to take it up and hopefully infer some sort of resistance to the coral. Adapting the algae. Here's another one. So it's well known, so the algae, there are actually hundreds of species of this symbiotic algae that can live inside of a coral. There's usually one dominant one, but there are hundreds of different partnerships that can happen. And some infer thermal tolerance and some don't. And so the algae you can culture in the lab, and you could expose it generation after generation to more heat stress and create thermally tolerant algae, and then inoculate a coral with it and hopefully get some sort of thermal tolerance in the coral. CRISPR. You guys have probably heard of CRISPR, or genome editing. This is actively being done by researchers just down the way at Stanford. So they're taking, this is a picture of some coral larvae that were injected at the embryo stage with a red fluorescent protein. And you can see these larvae producing red fluorescence, just demonstrating that the phenotype actually existed. And so now this same group is looking at genes that they think are implicated in the bleaching pathway and seeing whether or not they might be able to manipulate those to create thermally tolerant corals. Cryopreservation. This charge is being led by the Smithsonian, a woman named Mary Hagedorn. There's, we think, around 800 different coral species. Coral taxonomy is a bit of a mess. We think there's around 800. And so far, they have successfully cryopreserved sperm from 16 different species. So about 2% of the total biodiversity of corals, there is sperm banked at different repositories around the world. There's some in Australia, there's some in Hawaii, there's some in DC. We've talked about maybe becoming a site here one day, with the idea being that and they're working towards being able to cryopreserve eggs and larvae and eventually whole coral fragments so that you could use those later to reseed reefs. And just a couple of last things. We're not going to go through it all, but just to show you kind of the breadth of what people are talking about right now. This is a sun shield that's being developed in Australia by the scientists who developed Australian money, which is plastic. And what it is a polymer that's actually made out of calcium carbonate. It's biodegradable. And the idea is that you spray it over the surface of the ocean and it reflects sunlight. And so it reflects up to, I think, 30% of the sunlight, so that during a warming event, they're not getting the double stress of thermal stress and light stress. And then over time, it will biodegrade and hopefully shield the corals from stress. And the idea here would be that you have planes before a bleaching event fly over huge swaths of reef and spray this all over the reef. This is the crazy studies that I led a few years ago that resulted in a couple of Nature papers. And the idea was, can we neutralize ocean acidification? So we went to a reef in the southern Great Barrier Reef, and we took effectively soda lime, and we dosed a bunch of seawater upstream of a coral reef with soda lime to see if we could neutralize the pH and get the reef to grow faster. And it works. It completely works. But again, the issue here is scale and longevity. When you're talking about, this was an 800 square meter patch of reef over the course of a couple of hours. We're talking about global systems, Great Barrier Reef 2,300 kilometers long over the course of decades. So part of the problem with a lot of these, or part of the hurdle with so many of these potential solutions is scalability and longevity. Larval bots. This one's pretty cool. This one actually just got funded about a month ago by the Great Barrier Reef Foundation through an Out of the Blue Box challenge. And this is using underwater AUVs to collect coral larvae or collect coral spawn. And then you create larvae, and then you use these underwater AUVs to go over huge swaths of reef and just spit out larvae to try to reseed larvae. They're going to try this for the first time on Magnetic Island off the coast of Australia this fall, so stay tuned on that one. And then I'm really excited that this summer I'm going to be hosting a Fulbright scholar named Taryn Foster from Australia, and she's going to be here, and we're going to be working out of Cal Academy and with Autodesk, and her idea is to use robotics and 3D printing to try to scale reef restoration efforts. And so that's something that we're really excited about as well. Okay, so let's say that you don't actually want to tinker with it so much. You're a little bit maybe non-interventionist, and what you want to do is just kind of help give the system a boost, but you don't want to be making Franken corals. So how would you get more corals out onto the reef faster? The only way you can do that is by repopulation, which basically means sex. And that's where we come in at Cal Academy, and that's our focus, is trying to help facilitate corals come back naturally. And so in order to just put this all in context, I want to briefly take a minute to talk about how corals reproduce. Corals can reproduce asexually, which we saw at the beginning by budding within a single coral colony. They can also reproduce asexually by fragmenting. So much like a starfish arm, if you have a branching coral and you break off a branch of it, that'll regenerate and the original coral will regenerate. And around the world, people take advantage of this for reef restoration efforts. Corals also reproduce sexually, two main ways. Spawning, which is broadcast spawning. You may have seen, it's a very beautiful event that happens once a year when corals release eggs and sperm into the water column all synchronously once a year, and then they have external fertilization and make little larvae that then go and settle. And about 75% of corals worldwide reproduce sexually that way. And then there's a subset of corals that do what we call brooding, that actually have internal fertilization and make larvae inside and then release little baby larvae fully competent. So again, around the world, people are having coral nurseries, coral gardens to try to fragment different species and produce corals en masse to try to get more corals out onto the reef faster. This is an organization, Coral Restoration. They work in Curacao. There's several organizations out of Florida. And what you do is fragment them, grow them out on trees. You can see how quickly they grow, and plant them back out onto a reef. Over 90 species worldwide are successfully done this. We're really good at growing corals and getting corals back onto the reef. You can restore coral reefs on the area of a hectare. Problem is that destruction is happening much larger than that. And there's a really exciting new development in fragmentation that happened accidentally out of Florida a couple of years ago. The former president of Mote Marine Lab discovered what we now call microfragmenting, where if you break a coral, not an entire branch off, but break a little tiny piece off into a microfragment, because we think of some sort of natural healing response from that lesion, it will grow 20 to 50 times faster than a larger piece. And so what they do is microfragment them, and if they all came from the same individual, because they're clonal, when you put them back together, they fuse, just like tissue grafting. And so this is a procedure that's now being integrated into reef restoration efforts around the world. You can make coral colonies much larger, much more quickly. But the one thing that those fragmenting coral nurseries and gardens don't do is address genetic diversity, because what you're doing is propagating a single genotype. And arguably in a time of rapid environmental change, what we need on the reef is lots of genetic diversity. And so what we try to do here at the Academy through partnerships with nonprofits like Seacor, which I'll talk about in a second, is we try to facilitate sexual reproduction in corals naturally so that we get more genetic diversity out on the reef, more opportunities for success. So corals release, about 75% of corals spawn annually, one time a year, usually in late summer, after a full moon, right after sunset. They are surprisingly romantic. And they release eggs and sperm into the water column, and those fertilize externally, create larvae that then swim down, settle, and start to create corals. And a lot of places around the world, this process is breaking down because reefs have become so depauperate that you could imagine huge stands of coral that would all be really close together. They'd all spawn synchronously, and then everything's mixing, lots of chances for fertilization and success. Now you have these broken and depauperate populations. You might have a coral of species A up there, one down there. If they spawn together, they might never meet up, they might never fertilize, or the communication might be broken down such that this might spawn on night 1, that one might spawn on night 2. We see that all the time. And so there's never an opportunity for success. And so what we do is, I often liken it to coral midwifery, as we go out and we try to help them through this process to create more coral babies. And so we know when these corals are going to spawn, and we go out and we net them in nature. And when they do spawn, these little bundles will float up, they're positively buoyant, into these collector cups, and we can collect them and take them back into the lab. And these are tubes of a lot of different individuals. And so we have lots of opportunity for fertilization and genetic variation here. This is another example. These are all the coral eggs, and this cloudy water is the sperm that's been diffused into the seawater. So we separate the different gametes from different individuals, and then we cross-fertilize them in these huge batches of seawater. Like here, you can see we keep track of different crosses, and we develop coral larvae. In the lab, we know optimal conditions that don't include ocean acidification, don't include anything they're dealing with in nature. We can get 100% fertilization success in the lab, close to 100% settlement success, and we try to jumpstart them through these things that would naturally fail in nature. This is a coral baby that was settled onto a substrate, and then once they're to a certain point, these substrates are taken back onto the reef and planted so that they have a better chance of survival. And this organization, SEACOR, that we partner with to do this work, it's a nonprofit. SEACOR stands for Sexual Coral Restoration, has been doing this now for almost 15 years. And they actually have individuals that they reared in the lab, outplanted, that have grown up and spawned with their natural populations, which is really exciting. So one of the bottlenecks to this type of work is that you can imagine that going and planting individual corals on a reef is very labor intensive and very time intensive, because it literally takes, whether you're doing asexual fragmentation or whether you're doing sexual reproduction, you are taking individual coral colonies and you're epoxying them to the reef. And you might be able to do a handful of corals in a dive, and then you're trying to cover huge swaths of reef. So we're trying to figure out, through our partnership with SEACOR, ways to bypass that bottleneck and scale our efforts. So one of the partnerships that was forged is to try to create self-stabilizing substrates that we don't have to actually physically fix to the reef. That if you drop them, they will drop to the bottom of the reef and they will kind of stabilize themselves and not move very far. And you can imagine a scenario where if you had hundreds or thousands of these that had little baby corals on them, and you had divers that were going out and scattering them everywhere, that it would look something like this. You're just kind of scattering seeds in a field. And they've done trials, different species like different designs, and different reefs might have different preferences, because a healthier reef might have more topographic complexity, and a more degraded reef might be a little flatter. So you might want different substrate designs for different scenarios. And through a partnership with SEACOR and with Autodesk, there are some scientists here that are helping to move this potential solution forward. They've sent substrates now that were 3D printed here in the Bay Area to Australia. We tested some in our system to Curacao to try to see which ones the corals prefer. With the idea being that you would put hundreds or thousands of these out, condition them in the field, and then have these floating pools that you would settle thousands or millions of larvae onto, and then the divers would take them and scatter them onto the reef. Here's one of those substrates that has a little baby coral on it, and then a similar substrate, and it's hard to actually see the coral because it blends in with the reef so well, but this is the original substrate, and this is the coral that's grown over the substrate and into the reef. This is one potential solution. This work is currently being conducted in the Caribbean with focus areas of Curacao, U.S. Virgin Islands, the Bahamas, and Mexico, and they're expanding to different areas in the Caribbean and looking to expand into the Indo-Pacific right now. So now I want to talk to you about a project that I've been leading over the last couple of years that I'm really, really excited about. And what it is an idea to try to spawn corals ex situ in a laboratory setting. And this is not a trivial thing to do. And so what we're talking about here is developing protocols to create reliable and predictable reproductive events in an aquarium setting. So why would you want to do that? First of all, because you only have one opportunity for success in nature. These things reproduce once a year. If you wanted to test any sort of idea, whether it be CRISPR, you want to do genome editing, or whether it be testing 3D substrates, you have one shot a year. And if it doesn't work, you have to wait till next year. Meanwhile, we're losing a third of the Great Barrier Reef over a couple of weeks. And so if we can figure out ways to spawn corals in aquarium settings, then we have more opportunities for research, more opportunities for science and success. And we can kind of jumpstart a lot of these solutions. So we've been working with Jamie Craggs from the Horniman Museum in London, and he and his group are the only people in the world that have been able to do this, and they've been very heavily advising us. We've been working with them for about 2 years now, and we've had a lot of success, and we've done it 2 years in a row, and we're the only place in the US that has done this, and the second place in the world. So that's really exciting. And so what we're trying to do is mimic the hierarchy of environmental cues that will elicit coral spawning in nature. And again, because this happens once a year, there's a lot of things that we have to try to consider. We think that in some species, the process of oogenesis, which is creating those eggs, takes 9 months. It's usually late summer because that warming through the summer triggers maturation of eggs. Then it's after a full moon. Then it's after sunset. So if we're going to do it in an aquarium, we have to have a setting where we can really tightly control temperature and light, and numerous other factors to try to simulate seasonal cycles, try to simulate full moons and new moons, sunrises and sunsets, in order to try to get these individuals to spawn. And so we do that by using these microprocessors by a company that's actually based in the Bay Area. And that's one of the huge benefits of being here at the Academy, which I'll talk about again later, is that we are in this scientific and tech hub in the Bay Area that gives us access to things like this. So they actually were a donor for our project. They gave us most of the equipment in kind because they were so excited about what we were doing. And what we did was program in seasonal temperature cycles so that the corals experience a winter and a summer. And then we have lights that have 8 different wavelengths of light, and we can change the intensity of the lights, and we can change the distribution or of the spectra that they're experiencing to mimic different cycles that we want throughout the day or the month. And so what I want to show you now is a video that was made by our lead aquarium biologist, who's actually here tonight, just documenting the process over the last couple of years of the construction of this lab. It's a pilot project to see if we could actually do this. And I'm going to stop talking for a couple of minutes. This is amazing footage that Rich caught he's the lead aquarium biologist. This is the spawning in the tanks in the back of house of the aquarium in this building. We had a synchronous spawn of these individuals last year and this year. Our lab bench is currently a piece of plywood that my postdoc brought from his house. We were all a little punchy after this. The algae that started to get taken up. And that's one of the C-core tiles that was 3D printed. So, thank you. So we're thrilled at this point that we got this far already. We're in year 2 right now, and at every step of this, we had no idea if this was going to work. So we brought the corals. First of all, we didn't even know if the corals were going to survive on their trip from Palau, and we're all in there every day. These are our little babies, checking on them, making sure that they're okay, and they did great. And then we didn't know if they were going to spawn. And so we had a team of 3. It was my postdoc, Rich, who's here, and me. And we were checking every 15 minutes during this window that we thought they might spawn for about a month we're taking turns going in and just, are they going to spawn? Are they not going to spawn? I don't know. And then they did. And they spawned when they were supposed to. So we did a reverse day-night cycle because normally they spawn several hours after sunset in Palau. That is the equivalent of, I believe it was 4 a.m. for us here, and nobody wants to do that. And so we tried shifting the schedule to see if we could get them to spawn. So we have sunset in the morning for them. So it's a completely, it's like an old dark room. So it's completely light-proof room. And we simulate a sunset in the morning so that they should spawn around noon, somewhere between 11 and noon. And they did it. And it was amazing. And we were so happy when 7 of these colonies spawned synchronously last year. And then we fertilized. We got over 90% fertilization success. And then this year we had similar results, and we improved upon things as well. And so the idea now that we have some proof of concept is that if you can imagine a scenario where you could build a couple of these, right, and stagger them so that you have a spawn in the spring, like this. You have a spawn in the spring and you have a spawn in the fall. And then, or you could even imagine a scenario, and I don't know if any of us wants to do this much work, but where you have 4, and you have 4 spawns a year. And that gives you so many more opportunities for research and for restoration science. So just from this little spawn that we had last month, the babies that we have in the aquarium right now are about a month old. We had 3 different labs. We have collaborators from Washington, D.C., the Carnegie Institution for Science, from Stanford University, Hopkins, that were coming to us saying, we heard you have coral spawning. Can we get in on it? We have some things we'd like to try. And so we facilitated over 5 different PhD projects that were looking at everything from microbiome acquisition. The eggs are supposed to be released sterile. And so sampling at the egg stage, larval stage, to see how does the microbiome establish itself? Does it change? How does that influence overall health? We're actually doing a new coral genome because of this work. We're using coral sperm to create a new coral genome. We are looking at whether or not corals have germ lines and what that might mean for evolutionary biology. And we are looking at maybe next year trying a little bit of genome editing. So we have become this hub, and because, you might say, well, we're not proximate to any coral reefs. Why would you do this in the middle of San Francisco? But my answer to that would be that we're in the middle of one of the greatest academic centers with Stanford and Berkeley and all of the universities and great minds that we have right here, and then also the tech industries, but adjacent to Autodesk and everybody that we have access to, that we can get all hands on deck in terms of trying to find solutions to this. So that's that project. We're really excited about it. Please stay tuned. And then I just want to spend a couple of minutes just talking about how you can engage in this if you would like to. So if you've seen our Expedition Reef show, some of this is a bit repeat, so apologies for that. But really the way that, with 7.6 billion people on this planet, the way that we can all make a difference is by voting with, literally voting, so please vote, but also voting with your wallets and with the choices that you make every day. And so if you're not familiar with Seafood Watch, I mean, this is a science-based app about responsible seafood choices. There are some seafood options that are completely destructive to reefs and to oceans when they're taken out, and some processes and some types of species that are very unsustainably harvested. And so just consulting this when you choose any sort of seafood is a great way to try to make a difference. And then also, our planetarium team put together this analogy, which I love, which is the equivalent of how far you would have to drive in your car in terms of CO2 emissions if you were to choose between a veggie burger, a turkey burger, or a beef burger. And so if you did that once and it was lunch and you said, what am I going to have? If you chose a veggie burger, you would have to drive your car the equivalent distance of where we are just to Sunset. That's it. You're not going very far, a few blocks. If you drove a turkey burger, you'd be going down to San Francisco Zoo, somewhere maybe by SF State. And if you chose the beef burger, you'd drive all the way to the airport in terms of the equivalent amount of CO2 you'd be releasing into the atmosphere in terms of what it took to make what you were about to eat. And that's just one burger. But if you did it every day for a year, you'd be still in San Francisco, congratulations, or you would be in Baja. And you might want to be in Baja, but not for this reason. So, and it's not saying that everybody has to choose veggie all the time, but it's saying with 7.6 billion of us, if we can all just try a little bit harder to make responsible, sustainable choices, it will scale to make a difference. And the last thing that I want to say is that being in the academic and tech hub that we are in the Bay Area, that we need people as thought partners on this. If you have a skill or you know somebody that has a skill that you think would add value, have them come talk to us, because 1,000 coral biologists in a room in December in Florida talking about how to fix this is great, but we're 1,000 coral biologists. We're not engineers, we're not business people, we were all trained in the same way. And so what we need is cross-disciplinary thinking, people from different disciplines coming together to try to tackle this problem. And so we welcome that and we want your involvement. So please come and talk to us about it. So I'm going to end there, but I do want to thank all of the incredible donors that have supported the Hope for Reefs initiative and made it possible for us to do the exciting science that we love to do every day. So thank you.
I think we have time for a few questions, and I would just ask, we have 2 mics on either side of the room, and I would welcome you to speak your question into the mic. Thank you, Rebecca.
I'm sorry? How many species? We think somewhere between 600 and 800. Coral taxonomy is a bit of a mess because corals are very plastic in their morphology, and a lot of coral, the short answer is somewhere between 600 and 800. The long answer is that corals, that the same species that grows in a different environment, if it's deeper, if it's more wave-driven, will look completely different. And coral taxonomy classically was always morphological based. And that's being rewritten right now, because most of the time when someone will go and do a genetic study, they'll realize there are all these cryptic species that they thought were one species and it's not. And so the whole book is being rewritten, but we think somewhere between 600 and 800.
Quick question, just out of curiosity, what are the substrates made out of that you're putting the corals on? Is that like calcium carbonate, or what is the
They're
actual
trying
material?
a bunch of different materials. Some are cement. They do 3D printing in calcium carbonate. I don't know if they've actually 3D printed these ones in calcium carbonate, but they are trying a variety of different materials. All are non-toxic, making sure nothing's going to leach into the oceans.
With all this, have you thought about the law of unintended consequences when you are doing this? And have you come up with any detrimental effects that you can see, or foresee, I should say, regarding this?
Absolutely. There's all kinds of potential detrimental effects. And everyone's thinking about those all the time. And I would say that, just so some of the unintended potential consequences could be, let's say you create a supercoral and you release it and it outcompetes everything that was natural. No one wants that. The last thing that we want to do is anything like that. Generally, people take what we call the precautionary approach, which is we know what. The reason we're looking at all of these things right now is so that we have these things in our back pocket if we need them. Right now, you cannot release anything like this onto the reef. It's too heavily permitted, it's too heavily regulated. You can't release genetically modified organisms into the ocean. But people are allowed to do it in the lab so that if we get to the point that we need it, they might be able to. But that's on everyone's radar. We take a precautionary approach, which means you only do the next level of what you need to do at the time. You don't go very extreme. But I would say that what I've heard in terms of response is the community is a little bit polarized of what about all of the unintentional consequences. And then there are people that say, well, we know the consequence of not doing anything, which is we're facing 99%. And so there might be unintentional consequences, but would they be less bad than losing everything? And so that's another argument that's out there, which I'm not saying I condone or not, but it is part of the discussion, rest assured.
So you're welcoming people to talk to you if they're interested or interface, but what phone number or web address do
That
you
would be
use?
helpful. So
Oh,
my
okay.
my email is up there. Please feel free to email me.
Is there a place online where you can find your slides to your PowerPoint?
Question, are these going to be publicly available?
How does sunscreen with oxybenzone play into the whole situation?
That's a great question too. So there's been a lot of media attention about the negative impacts of sunscreens and choosing reef-safe sunscreens. And so we do know that certain chemicals in certain sunscreens are negatively impact corals. They lower growth rates, they lower survivorship, and they lower, on early life history stages, they are particularly susceptible. So coral larvae, there's higher mortality with them. And I know that they are trying to get people to choose reef sustainable sunscreen or not wear sunscreen at all and just wear SPF clothing. In Hawaii, I believe they've made sunscreens with oxybenzone completely illegal. I think they're trying a similar campaign in Fiji or in Palau right now. So that's starting to scale. So yeah, thank you for that question. I think it's interesting because this came up last year at a talk, and someone said, well, is that really the solution to the problem, not wearing sunscreen when it's really a global warming issue? And the answer that the speaker gave, which I really liked, was no, it's not the solution to the problem, but she likened it to, like, also plastic straws, a gateway drug to conservation, because what we need is 7.6 billion people that care. And you're not going to get 7.6 billion people to convert to veganism and go start a coral nursery, but you might get 7.6 billion people to change the sunscreen that they choose, or maybe not opt for a plastic straw. So kind of a lengthy answer to your question, but thank you.
As a pediatric dermatologist, I will say that UV light does lead to skin cancer, and not using a straw doesn't. So I am very pro doing anything that we can to protect the reefs, but there's a lot of things we can do besides stop using sunscreen. Although I really like physical blockers, mineral sunscreen too. I was just curious, what is the equivalent FDA for putting plants out in the ocean?
Putting plants or putting coral?
Well,
or just
coral.
putting things into the ocean.
For FDA approval
Sure.
for drugs in humans versus
It
coral.
depends
and
on where
ocean.
you're talking about. And so it's whatever regulatory agency is responsible for that particular area. So in Florida Keys, for example, it's the Florida Keys National Marine Sanctuary. There are local and regional regulatory agencies. On the Great Barrier Reef, it's what we call GBRMPA. It's the Great Barrier Reef Marine Park Authority. And you have to submit a permit application. It's very heavily vetted. It usually takes almost a year to go through, things like that. So it depends whether it's a local, a regional, or a national governmental agency, but it's usually government and some sort of conservation agency simultaneous that evaluate requests.
As the oceans are warming, is the coral developing in parts of the ocean that are now at a temperature that's sort of more in the range where they historically inhabited?
Is there any sort of migration?
Yeah.
Yeah, there is some evidence. So there's evidence of reef fish migrating polewards to higher latitudes. There is some evidence that certain species of corals are migrating polewards. On the Great Barrier Reef, there's a little bit of evidence that there might be some species that are found further south than they were before. it's a little harder for corals to do that because they are benthic and they are fixed to the floor. And so the only way they would do that is through dispersal of larvae. And then that larvae has to settle and grow. So it's a much longer time than a fish just moving around. But there is a little bit of evidence of that happening. The problem with it, though, is that the combined stressor of acidification. So the higher latitudes, the more poleward you get, the cooler the waters, but the more corrosive they are. So now you're edging more into ocean acidification territory.
I just wondered, could you explain the difference between hard corals and soft corals? And are they completely different organisms, or do they interact in some way?
Yeah, that's a great question. I wish Gary was here. Is Gary here? Gary's our soft coral expert. So hard coral, that's a great point because I was speaking about hard corals exclusively this evening. There are actually more species of soft corals. Hard corals are scleractinia, and they are corals that secrete a calcium carbonate skeleton underneath them. Soft corals do calcify inside the tissue, but they don't actually have a calcium carbonate structure that's underneath them. So when you're talking about a coral reef that's accreting over time, most of that calcium carbonate is from hard corals and some calcifying algae. But they do interact. There are some reefs, like in Fiji, that are mostly dominated by soft corals. But there are, on a lot of reefs, there's a mixture of soft corals and hard corals together.
Join me again in thanking Dr. Albright.